Hello and welcome to Lecture 4 of the MOL213 Developmental Genetics course. My name is Fergal O'Farrell and I'm here to take you through it. And in today's lecture we will continue our work on understanding patterning in Drosophila. If we have a look at the learning goals for today's lecture, we will, as I said, continue with pattern formation, but we will be looking later in Drosophila development. We will also learn some genetic techniques. I've mentioned the p-element transposons, and we'll have a brief introduction to that, as well as other interesting techniques, which include the binary expression system UAS-Gal4, which is used a lot in fly genetic experiments. And we will try to understand how we perform a mosaic analysis by mitotic recombination. We will also meet a couple of signaling pathways in a little bit more detail, the Wnt and the hedgehog signaling pathways, and we will try to understand the processes of segmentation and compartmentalization. And then we'll make our first foray into the world of Hox genes and try and understand how Hox genes set up and maintain segment identity. Okay, let's get started, shall we? So let's say a few words about p-elements. P-elements are a very useful tool in Drosophila. What is a p-element? It is a tool that we have generated that is derived from DNA transposons. And DNA transposons, I've shamelessly lifted this from Wiki, are a DNA sequence that can change its position within a genome, sometimes creating or reversing mutations and altering the cell's genetic identity and genome size. Transposition often occurs in duplication of the same genetic material, and Barbara McClintock earned a Nobel Prize in 1983 for her discovery of them. So what does that mean? So it's a DNA sequence that can copy and paste itself around the genome. Okay. Sometimes when it lands in a new position, it can create mutations. We can use it as a tool, perhaps, to reverse mutations. Yeah, and so it's usually copy, paste, duplication of the same genetic material. These transposable elements, as they are called, these DNA transposons, are found in almost all life forms, and their origin and evolution is being examined, as is their effect on genome evolution. So a large amount of our genome is actually composed of transposable elements that are no longer jumping or copying and pasting around the genome. And they usually end up in a sort of a scrapyard for these transposable elements close to the centrosome there, where you have this repetitive DNA that's often composed of used transposable elements. But there are still some jumping in our genomes, and these are not good for us in general. Most are regarded as selfish DNA parasites, and in this way are similar to viruses. This is also lifted from Wiki. I won't go through it all, but just mention that they can cause diseases, and one of the most famous ones is Alzheimer's disease. So if you want, you can pause this and read through it yourself if you're interested. For us, the most important thing is, how does it work, and how can we, if we know how it works, can we use it then to our advantage? So the very typical transposable element, this is a Mariner type here, has a DNA stretch of sequence that encodes for just one gene, and that is a transposase, a DNA transposase that recognizes specific sequences on the transposon and can, when it's produced by the genome, that's why in that way it's a parasite. It relies on us to produce its transposase protein. The transposase protein then recognizes these elements flanking the transposase DNA and will actually cut it out, lift it up, and allow it to insert in In this case, it says as a specific motif, but some P elements jump randomly or randomly with bias. And they've been studied. There's different types of this sequence that is recognized by the transposase, and they have different types of specificity when it comes to inserting into the host DNA again. Many times it's in the 5' region of a gene just before the transcription start site. And in that way, it can have negative effects because it will lower the amount of wild type gene in the genome that is transcribed and steal some of the DNA polymerase activity away from the neighboring gene to itself, to produce itself. And then the real sort of risk is that these inserts and these excisions create mutations in genes, which can happen, but it can also create a lot of mutations in the promoter region and affect expression of these genes. Because when they jump in, they cut themselves in, but when they jump out, they leave a little bit of themselves behind everywhere they go in every jump. So these things are considered quite dangerous for the genome. So how can this be of use to us in flies? Well, people working with flies were very smart, and they separated actually the transposase from these transposase inverted repeats, or inverted repeats we'll call them. Target inverted repeats, maybe they're called, I can't remember. So they separated these away, and they took the transposase gene and put it on a different DNA vector, right? So now these repeats in the genome wouldn't be able to jump, let's say. But what they did was they took these repeats and put them on another vector. So the transposase encoded onto vector A, let's say, and the repeats recognized by the transposase put onto vector B. And that's what we see sort of here, I think, in this picture. Yeah. So and then the vector, the DNA vector that has the inverted repeats, will be inserted into the genome when it meets the other vector encoding the transposase. And one of the cool things then is that we can put whatever gene we want to within a certain, there are certain size limitations, but we can put genes, say up to 15 kb, inside the vector with the P repeat elements. And then we inject the embryos, typically inject the DNA into the pole cells of the embryo. And remember, the pole cells are in the posterior part of the embryo and will generate the germ cells. And then we let these eggs, the ones that survive the injection process, they come up to adults. A good injectionist or technician who's good with injection, you know, they will have over 90% survival of the eggs will make it to adults. So we inject into the pole cells so that these adults then, their gametes, can produce progeny where there is stable insertions in the germline. And then we have to screen in the next generation to look for stable insertions. In this example, they've put back the white gene, and if you remember, white mutated flies have white eyes. The normal eye color would be red. They put back the white gene, which will give a red eye color in successful transformants. I hope that's clear. So what you've generated then is a useful tool with visible markers. You can also introduce other genes here, for example, ampicillin resistance, so that you can then cleave out the DNA, you crush up the flies, cleave out the DNA, you have ampicillin resistance in your vector, you can digest out your vector from the DNA, recircularize it, and transform bacteria. And when you do that technique, the normal thing is that you recover flanking DNA from the animal's genome, and you can sequence that to see exactly where your P element has been inserted. And the ones you'll be interested in would be the ones that actually give you phenotypes, right? So you can use this as a way to screen for genes that are important during development, because these insertions, as I mentioned, they can affect gene expression, and that can give us phenotypes then that we can screen. So, and this is mentioned here. The technique can be used to increase the number of copies of a particular gene, So you could put back, if you have a mutant generated by one type of mutation or another, you can put the wild type gene in this vector, inject it into that mutant, and you can see if you can rescue the phenotype by putting back the wild type gene. Or you could overexpress a gene this way if you wanted to as well. You can put back the mutated gene with alterations in the coding region, and in this way you can figure out if molecular regions of the protein that the DNA encoded for are important, right? So you can do detailed molecular analysis using this approach to get wild type or mutated genes back into the various genetic backgrounds you're interested in. And then you can introduce new genes. I mentioned this with, for example, ampicillin, but you can also put in reporters like LACZ, which you can use beta-gal to stain for the expression of the LACZ gene then, or GFP or gal4 coming from yeast. And then the insertion site, as I mentioned, is typically in the 5' area of the gene, stealing some activity from the wild type transcription of the neighboring gene. And in this way, we can get a reporter, right, for the gene expression inserted into the Drosophila genome. This was very popular in the 80s and onwards. It was used in mutagenesis and in generating reporter lines for certain genes. And nowadays we have CRISPR-Cas to do these things for us. So this is a slightly older technique. It is still quite useful because you have these P element collections now inserted in all genes in the fly. We have maybe 5 times coverage of the genome. So if there's 14,000 genes, we have maybe 80,000 P element lines that you can request, and either they have a phenotype already, or you can then introduce the transposase again to these animals and get the P element to jump out. And what happens when it jumps out is that most of the time it jumps out correctly, but as I mentioned, sometimes it makes mutations, and useful mutations for us is that it deletes areas of the genome around it. This happens in rare frequency. So the excision of the P element from the genome isn't 100% accurate. It can make deletions in neighboring genes, and that can be very useful for us to make new mutants. And this enhancer trapping is just basically what I mentioned here with the LACZ. They call it enhancer trapping, that you can put the LACZ in with a minimal promoter in front of it and then land in front of genes and steal polymerase activity, and you'll get a reporter. So this brings us to the gal4 UAS system, and this is a binary expression system where gal4 is a transcription factor originally from yeast, and the gal4 transcription factor binds to the upstream activating sequence, UAS sequence. And this is shown down here. We have gal4 gene gives rise to a protein, binds to these UAS sites, and this will then drive expression of a gene that is tagged with this UAS promoter. And this can be coupled with the P element system, where we have these inverted repeats on either side of the gal4 transcription element, and we can then jump this element around in the genome of the flies, and we will get insertions all over the genome that will express gal4 in a certain gene specific pattern, different in each one depending on the landing site. And this has several uses. We can then see where these are expressed by crossing to UAS GFP, for example, or UAS LACZ, and see where these gal4s are expressed in time and in place during development, and then use these lines, these gal4 lines later on, to give targeted gene expression to figure out the function of genes, for example, or very useful in recent times is UAS tagged RNA interference lines, so RNAi. Then we can do targeted knockdown of genes in various tissues at whatever place in time. So some of these, as I said, use P elements to insert in the 5' area of a gene, and they will steal the enhancer promoter expression of the gene they sit next to give a gal4 expression. But we can also put promoter and enhancer regions in front of the gal4, and then put these inverted repeats on that DNA construct and put that into flies, and then we will get a predicted expression pattern. Or we can figure out, use it as a way to figure out where that promoter and enhancer is expressed. So in this way, it gives us tissue-specific expression with some things, or even cell-specific expression. In the case of nerves in the brain, for example, there's a lot of Gal4 lines with specific expression in different nerves. And then we can express whatever target gene or RNAi knockdown constructs we want in those cells at those times, targeting what proteins we want. So this is a very powerful tool. And the typical thing is that we have hundreds of these lines with Gal4 expressed in different patterns. And in the case of RNAi lines knocking down, we have thousands of these lines. So there are multiple RNAi stocks of flies available with UAS RNAi to cover every gene. There are multiple lines, usually 2 to 3 lines for every gene on average. So we have 14,000 genes. So we're talking about 45,000 RNAi lines that are available. So it's, if you want to study the function of a gene, you can get those RNAi lines and get a Gal4 to look at that is expressed in a certain time or place, and look at the function of your gene at that time and place, for example. Okay, back to developmental biology here now. So we rounded up the last lecture by discussing how dorsal ventral patterning was performed. We'd already covered how in the egg you set up anterior, posterior, and dorsal ventral axis, and then we discussed the molecules, for example, DPP, that was involved in the dorsal ventral patterning. And we need then to discuss how the anterior posterior patterning is set up to overlay the anterior posterior axis. And we discussed that in the egg we had deposition of, for example, bicoid anterior position, and that set up gradients, right? And we mentioned that some gap genes controlled, for example, hunchback expression was controlled by the levels of bicoid. And we've discussed that enough, but we can now mention the other gap genes. And the gap genes are the first zygotic genes that are expressed along the body axis. And all of these gap genes encode transcription factors. Most are zinc finger type transcription factors. And their expression pattern is defined by the miternal bicoid gradient to some extent, and by gap gene interactions. And that means that the expression of these will restrict each other, is the best way to say it, I think. So, and hunchback is an important regulator of the other gap genes. And these gap genes are required to set up a periodic or sort of repeating pattern of transcription factors that delimit broad body regions. So they're important to define these broad body regions, and the mutant phenotypes will show gaps in the anterior posterior patterning. And as these pictures show, there's a gradient of expression. You can see that it's strongest in the middle, weaker on the flanks, strongest in the middle, weaker on the flanks, for example. And the gradient of gap proteins are established by diffusion in the syncytial blastoderm, and in general they are short-lived proteins, these transcription factors, and this gives rise to steep gradients. So they are produced in a certain place and diffuse out, but they don't live long. So they give it's already, the protein is turned over at this region here. So you get a steep gradient, right? And then the key function of the bicoid and gap protein gradients is to initiate the expression of the pair rule genes. So these control the expression of the pair rule genes. And the pair rule genes then will define the power segments. And we've discussed this already, that bicoid, the gradient of bicoid controls the zygotic expression of hunchback, and we talked a good bit about how a certain threshold will trigger expression of hunchback and give this sharp border where the levels of bicoid protein have dropped down and are at levels below the required amount to trigger transcription of hunchback. And we also mentioned that Nanos was involved in the translational control of hunchback. These together gave rise to a hunchback protein gradient. And this slide we've seen similar before. It's just to remind us that we have this very sharp response, even if the gradient sort of drops off fairly smoothly. There is a sharp response to that. We've discussed this before, and the lecture by Eric Weishaus that I put up covers this extensively. So if you have more questions on this part, well, check out those lectures by Eric Weishaus. For now, let's look at another gap gene, CRUPL, and CRUPL is activated by low levels of hunchback in the presence of bicoid, but it is repressed at high levels of hunchback. So in a way, it's activated between two thresholds of hunchback. This is shown here. We know hunchback was broadly expressed and tapered off around here. And we see that as the level of hunchback comes down, there's a repression when the signal is high, and then we can have this domain of hunchback level where we get activation of CRUPL transcription, but as the hunchback gradient drops down, it's not sufficient to activate it, right? So we get this stripe of CRUPL. And this can be demonstrated to some extent. If we have a bicoid mutant, we have low levels of hunchback, and CRUPL will be expanded. The CRUPL domain will be much more broad, right? Just due to these lower levels of hunchback, quite a lot of it sits in the activation domain. Hunchback also represses other gap genes, KNERPs and giant, and again in this it has these specific thresholds of hunchback that will act to sharpen the anterior expression borders of expression of KNERPs and giant. And then between the gap genes, the other gap genes, we have cross inhibition. So we will have a peak of CRUPL, and it will interfere with the expression of other gap genes where CRUPL is expressed. It will suppress them, right? And this will sharpen and stabilize then these expression domains. So we have these gradients of maternal transcription factors like bicoid and caudal, and to some extent hunchback has some maternal contribution, but there's also zygotic hunchback, and these gradients then can be interpreted by the enhancer regions of these gap genes, which also includes hunchback, and then this interpretation will give rise to the stripe expression like of the gap genes. These gap genes in turn will regulate the expression of the pair rule genes, which will give us stripes of expression to define then what we've referred to before as the parasegments. These parasegments are very important as a preliminary way to set up segments in the animal, to establish the segments. And it's actually segmentation genes that will then define the positional values of cells within each segment. This is accomplished by cell-cell signaling, and we hope to cover all of that today. I can remind you about the parasegments and segments. The first thing to think about is that the parasegments are sort of temporary segments that are out of register with the final segments. And what do we mean by that? Well, each individual parasegment has an anterior region and a posterior region, and then when the final segments form with these very defined grooves, we see that the anterior region will be the posterior region of a segment. The posterior region of a parasegment would be the anterior region of the final segment, and the parasegment function as a sort of signaling center to organize the final segments that will form. So the segments and the parasegments use different compartment borders. But the segments that actually form in the late embryo, they will persist into the larva and even the adult. So what genes are important for segment forming Drosophila? Well, right from the start, there are 3 groups of genes that are being grouped according to their phenotypes and the stages at which they act. And then in the beginning, at the start, we have the gap genes, where mutations cause deletions of regions, including several segments. We have the pair rule genes, where mutations cause a series of deletions affecting alternate segments. And then we have the segmentation genes, or segment polarity genes, which mutations of cause deletions of a part of each segment, which is replaced by mirror image duplications. And all of the gap and pair rule genes are encoded by or encode for transcription factors that have been distributed in the syncytium. And in this figure, we see the expression in green in a normal first instar larva of the gap gene couple. And when it's mutated, we see that we lack many of these regions, including the head regions. In the pair rule gene, called even skipped or Eve, we see that it is expressed in these stripes, and when they are missing, we lack all of these regions in the first instar larva. And the segment polarity genes, for example, one called gooseberry, which is expressed in these one side of the segment, and when it's gone, we get this sort of mirror image kind of transformations of each segment. So the gap genes, setting up these broad stripes, they function to turn on the expression of the pair rule genes. And Eve, even skipped, or Fuji Tarasu. And here we see the expression of Eve in sort of blue-black and Fuji Tarasu in brown. We see that the head and tail regions fall outside of this pair rule gene expression, and we will examine how, for example, a head can be made a little bit later on. So these pair rule genes define the anterior boundaries of every parasegment. So there's 14 parasegments. You have 7 Eve stripes, 7 Fuji Tarasu stripes. And these start to be expressed as well at the syncytial stage. And as we said, the mutations affect the alternate segments. And this begs the question, how are the stripes of expression established? And we know they're going to take cue from the gap genes, right? So when we look at the expression of the pair rule genes, the first thing we notice is that early on they have quite broad stripes, and this becomes restricted to more narrow stripes over time. And these stripes are known to be sharpened in this way by an autoregulatory loop. So the protein, for example, even skipped, binds to its own gene when produced and increases transcription. So you will have no enhancement of the transcription at the edges of the stripes where protein levels are low, but you will have increased transcription at the center of the band. And each stripe is regulated independently, and the localization of each stripe depends on separate regulatory modules or enhancers in the even skipped and Fuji Tarasu genes. We'll have a look at that next. So if we look down here, we have a schematic of the even skipped gene region with the coding region shown here in red, which gives rise to the even skipped mRNA. And what we see in yellow here are mapped out regulatory regions, so enhancers that are both 5' to the gene and 3' to the gene that control its expression. And these enhancer regions they react to different combinations and levels of the maternal and gap proteins. And they will then, remember, we have these gradients of these maternally contributed proteins, and then they drive the expression of the gap proteins in these stripes. And within the gap stripe, there will be the right amount of transcription factor, or the right combination of transcription factor to activate one of these regulatory modules and drive transcription of Eve in that corresponding pattern. And this schematic shows which enhancer actually corresponds to which stripe. And these experiments have been done where they basically chop up the DNA in the region surrounding the coding region, and they fuse that region more or less blindly in the beginning to reporter genes, for example, LAC-Z, and then they put that transgene back into the animal and see where it's expressed. And then by refining those fragments that they started with, for chopping off bits and pieces, they can see exactly what DNA coding or what DNA non-coding region they can map the enhancer to the expression patterns. So in these animals we have the wild type Eve expression, and we see it marked with an antibody in orange, and then we have LAC-Z reporters containing fragments of DNA, and not the Eve gene, but the LAC-Z gene instead, and we can see where it's turned on. So when they have such reporters that reflect the expression of a domain, they can perform very detailed molecular analysis. And stripe 2 module or enhancer region of even striped, it's about 500 bases long and contains binding sites for picoid and gap proteins. And some of these Eve stripe enhancer regions are the most studied DNA up to now, I would say, with really intensive mutational analysis to map what nucleotides are important for expression, right? And then when they map the areas that are required, they can find out what transcription factors are binding there, if it's already known on the basis of sequence, but otherwise they can perform DNA protein interactions and figure out the transcription factors that are required. In the case of stripe 2, it's known that bicoid and hunchback act as activators, whereas the other gap genes, giant and crupple, act as repressors and then define the boundaries. This is shown here. We can see Eve stripe 2 of expression. Bicoid is at a certain level in its gradient and hunchback levels are high, while the repressors, crupple, are strong outside of this parasegment 3, but the levels drop off here, allowing for Eve expression. Similarly with giant, we have a peak of giant in parasegments up to 2, drops down by parasegment 3. And this combination of factors binding to the DNA, these activators and repressors, then allow expression in this defined domain. And here is an example of the binding of different transcription factors to one of the stripes of even skipped. We can see that there are activators and repressors, and in some cases it may be that the presence of a repressor actually blocks the binding of an activator. They're competing for the same site, for example. So these modules read the concentration gradients, right? If you think about it, I mean, stripe 1 here, for example, it's obvious why Eve can only be expressed here, right? We have repressors in the other areas that block its transcription, but the Eve gene is the same, and the enhancers are the same in area 1, but it will be other enhancers that are insensitive to levels of giant, for example, that will be turned on. The other enhancers don't have for the other enhancer that will be expressed in parasegment 1 does not have a binding site for giant, for example, right? But it has binding site for hunchback and bicoid, and it can be activated there. There are then other repressors that will prevent it from spreading to parasegment 2. Hope that's clear. And to complicate matters further, some enhancers, for example, so the enhancer that gives rise to stripe 3 interprets hunchback as an activator. The enhancer that gives rise to stripe, that was stripe 2 at parasegment 3. The enhancer that gives rise to stripe 3 at parasegment 5 interprets hunchback as a repressor. So its enhancer region is repressed by hunchback. So only when hunchback levels drop down low can we get a stripe of E expression here. And this can even be seen then if we look into the embryo, we see that hunchback in red, its levels drop off here, and then we can get this expression of Eve coming on. So it's complicated, but it's a really good example of how enhancers control expression of genes and how enhancers enable genes to respond to levels of morphogens surrounding the cells, right? So the enhancer can take positional information. Here I should be expressed on the basis of the gradient, the level of expression of various transcription factors, and uses this then to establish patterned expression. So we've seen how the gap genes set up expression of the pair rule genes to generate the parasegments. And now we want to see how this temporary pattern of pair rule genes can be used to establish expression of the segmentation genes and define the segments in the animal. So these segmentation genes, they're activated in response to pair rule gene expression, and they're expressed in a subset of cells within each parasegment, 14 stripes for each gene, so 14 stripes, 7 for each gene. And the segmentation genes themselves are a diverse group of genes, but yes, there's transcription factors there, but also proteins that encode for signaling, cell signaling pathways. And the mutants then have defects in each segment. The engrailed transcription factor plays important roles in segmentation. It's a homeodomain transcription factor, and it works together with Hox genes, which we'll hear about later on. It also plays a role in the developing central nervous system. You might hear more about it there. For our purposes, we refer to it as one of the segmentation genes, and there it has several functions defined here. It sets up a boundary of cell lineage restriction, and what we mean here is that it sets up a border, so a borderline. If we look here in this picture, we see engrailed expression is the round dot in the nucleus. This means that it's the transcription factor is in the nucleus and controlling gene expression. We see it's expressed in both Fujitaratsu and even skipped stripes. And these Fujitaratsu and even skipped stripes, they set the parasegment boundaries. Back to this point now, engrailed sets up a boundary or a border where it's expressed that restricts cell lineage. That means that cells that are not expressing engrailed cannot move through the border or mix with the engrailed cells. So yeah, it defines subdivisions, compartments within each parasegment. It defines a compartment, and a compartment is a group of cells with the same transcriptional profile that act together. By that I mean they will differentiate together. They have the same identity, that they form a compartment. Okay, so there within this parasegment, engrailed is forming compartments. It acts as a selector gene, specifying the identity of cells within that compartment. So not only does it define that, it marks that compartment, but it makes that compartment. It's not just a marker for the compartment, but it maintains the identity within that compartment. And these type of genes are often referred to as selector genes. There are not too many examples of this selector gene, but you'll probably meet a few in this course. And then in addition to these roles, they help to stabilize the parasegment and segment boundaries eventually. So in an engrailed mutant, you would have fuzzy definition, not a sharp definition between the parasegments, and similarly, not a sharp definition between the segments. You would have problems forming defined segments, right? So and that's on the basis of these roles, right? It prevents cell mixing, it defines the anterior end of the parasegment, etc. And initially then, engrailed is expressed in a single line of cells at the anterior margin of each parasegment. So here we have the parasegment boundary, and it's on the anterior side within the parasegment. Okay. The next stripe is the anterior side within the next parasegment. And we said that the parasegments initially, when we look at even skipped and Fujitsuratsu, they're not sharply defined, but they became more sharply defined through self-auto-regulation. And the engrailed expression in one single cell line in the beginning results from a combination of not just even skipped and Fujitsuratsu, but other pair rule transcription factors that help define it down to one cell layer. As the embryo grows, actually, it will expand slightly. So this just repeats. Engrailed is expressed at the anterior margins of parasegments. It makes a boundary, a border that prevents the mixing of cells, and this will make sharper or straighter parasegments. And if we look at engrailed expression within situ in the developing embryo, shown here, we can see that its expression correlates well with visible parasegment boundaries that are these transitory grooves, small indents that will not be maintained later in development. But in this is showing that engrailed marks them on the anterior side. This is just curiosity here. It's not going to be on the test, but the first engrailed mutant was actually found in Norway. We can have a look at this later. And then it was characterized much later than in the 80s by another Norwegian guy, Anders Fusa, who used to give this course, but he's retired now. And these experiments defining the role of engrailed, for example, in cell lineage restriction, forming this border, have been done using cell lineage studies via a technique that I'm going to detail later. But for now, the important thing is that they make clones of marked cells with, for example, you could have GFP, randomly mark these cells early in the embryo, note the position, and then look at them later on in development and see how they grow. And the marked clone grows into a bunch of cells here, and this cell grows into a bunch of cells here. And they could see that the engrailed expression, as it came on then later, they could see that all the cells descending from this clone stayed engrailed and did not mix with this neighboring compartment or cross over this anterior border of the parasegment into the next cells. But if they mutated engrailed, performed the same marking experiment, then this cell, for example, would the clone as it grows, so every cell that derives from this is marked with GFP as well. Without engrailed mutants, then some of these cells passed over into the next compartment. So we had green cells here and here in the absence of engrailed. So engrailed's role then was to make this border. And we'll see why that is important to keep this border there in place when we want to make the segments, and how important it is for cell differentiation later.